Good afternoon, everyone. Thank you for joining us for another of our Saints Talks, short presentations and lectures by leading members of our academic community on subjects that we hope of our general interest. My name is David Williams and I look after the alumni and supporter relationship with St Andrews, and I'm delighted to be able to introduce you today to Professor Gareth Miles. Professor Miles did his PhD in Auckland, New Zealand, and then did his postdoctoral work at Dalhousie University in Canada. That's a university that's impossible to find the correct pronunciation of when you check it online. He's now Professor of Neuroscience and Head of the School of Psychology and Neuroscience at St Andrews. His research is into the neural control of movement in both health and disease, and his basic research is about deciphering motor circuits of the spinal cord and the brainstem. In terms of the biomedical side to his work, it's about the disease mechanisms underlying devastating neurodegenerative disorders such as motor neuron disease. Professor Miles will talk for about 20 or 25 minutes and then we'll take questions. You can ask a question by typing that into the Teams chat bar. Please don't put your full name or any contact details because we don't tend to publish those for data protection reasons. We'll aim to finish about 10 to 6. And now over to Professor Miles. Professor, thank you.
Great, thank you very much for that kind introduction, David, and thank you to people for coming along to listen and maybe for listening back to this. It's a real pleasure to have this opportunity to talk to you all about some of the research we're doing. I thought I'd start with this cover slide, which is a beautiful illustration drawn by a former student in the lab, Vanya. This shows some of the key elements that we find within the nervous system. So today I'm going to be telling you about how some of these key elements help to control our movements. And then I'm going to tell you a bit about how some of the changes in these elements might contribute to some devastating diseases with a particular focus on motor neuron disease. So I've been interested ever since I started studying neuroscience in New Zealand in how the nervous system controls muscles and therefore our movements. So this includes things that seem fairly simple, like posture. We need to get our body in the right position, onto more complicated movements such as walking or locomotion, which is a stereotyped rhythmic pattern of muscle activation, and then onto more complex movements which involve many different muscles all coordinated in a careful sequence. So all of these movements, whether it be posture control up to the complex goal-directed movements, they all involve our nervous system. They're controlled by our nervous system, which you see here. Now, when we talk about the nervous system, we tend to think about the brain, but there are other parts of the nervous system that are very important and are doing a lot of the work too. And that includes areas like the brainstem and spinal cord. So in fact, it's the spinal cord, which is running down our back, of course, that I have focused on for the bulk of my career to date. And that's because the spinal cord actually controls a lot of what we do. It's not just a series of wires connecting the brain to the body, but there's some complex things controlled within the spinal cord. So how do we know this? Some of this knowledge has come over 100 years ago. A good example is work done by Graham Brown back in 1911 to try and understand what parts of the nervous system control walking or locomotion. And back in this work in 1911, Graham Brown showed that if the brain is no longer properly connected to the spinal cord, and this was done in quadrupedal four-limbed animals, you can actually still get activity that would control walking behavior in the hind limbs of these animals. So these are otherwise paralyzed hind limbs with no longer getting input from the brain, but on a treadmill you can retrain these hind limbs to walk again. And what this shows us is that within the spinal cord in isolation, we have what we need to control walking. Of course, this has important implications for things like spinal cord injury, and a lot of people around the world are looking at how we can use this knowledge to try and repair or re-establish walking in people who've had injuries. So this is true in multiple species. One important question is is this true in us, in humans? So this is an example of what's often termed a stepping reflex. This is my son about 15 years ago. He's a few weeks old. If you can get your hands on a baby, ask the parents first. This is an interesting thing to try. If you put that very young baby on a flat surface, tilt them forward a little, you might get this rudimentary stepping activity, the stepping reflex. So what's interesting here is that this is at a stage when the descending inputs, the systems in the brain, haven't fully connected to the spinal cord yet we can still see this rudimentary stepping. So this is some of the evidence that we believe this is true in humans as well, that we also have these control systems embedded within our spinal cord that actually control our walking. So what are the things in the spinal cord that are doing this? So there are many neurons in the spinal cord. This is a cross-section looking into the spinal cord, showing the many neurons in a very thin section of spinal cord, these red spots that hopefully you can see. So what are some of the key neurons within the spinal cord? Well, we have our motor neurons. These are large neurons that connect to the muscles, and all of our movements are controlled by motor neurons, which make the muscles contract. We then also have what we call interneurons, lots of other neurons within the spinal cord, and they form these important networks, these teams that control some of our movements. We have information via sensory systems which come from the skin and the muscles and the joints, which tell our spinal cord and indeed the brain what's going on in the world, what's our body doing, what's going on around us. We also have connections from the brain, of course, that are important to control what the spinal cord is doing as well. And of course, there are many of these neurons all jam-packed in. So a major challenge is trying to understand how they come together to control our various behaviors. So what we know from the work of people like Graham Brown and many others after that is that we have what's called a central pattern generator, or a CPG. This is a group of neurons in the spinal cord that work as a team, as a network, and they produce the rhythmic alternating left-right activity that we require to control walking. And that's true in many, many animals. It could even control swimming or flying, but there are these similar central pattern generators embedded within the spinal cord that do this job. So how do the neurons in their work, how do they do this job? Here's an image of a neuron, an actual motor neuron. Here's my stylized version of a neuron. And we have the neuron here. We have this part we're pointing at here is the cell body. And then we have this wire-like process we call the axon. And this is the path by which this neuron forms connections with other neurons. And we call that connection, where the two neurons meet, we call that a synapse. So neurons work by generating and passing on electrical signals. So these electrical signals travel down this wire, this axon, and we call these electrical signals action potentials. The question then is, what happens when they get to the synapse? So what you'll see here is that there's a gap between the neurons. And this is where some important molecules called neurotransmitters come in. So these neurotransmitters allow us to bridge this physical gap between the neurons. And they do this by signaling via things called receptors on the neuron that's receiving the information. And these are specific to the neurotransmitters. So when this electrical signal, this action potential, travels down the axon, this causes the release of these neurotransmitters. They bind to these receptors, and that then causes an electrical response in the next neuron in the chain. So we have an electrical signal, a chemical signal, and then an electrical signal. And this is the way that these neurons talk to each other throughout the brain and the spinal cord in these complicated networks. However, it's not just neurons that are there. There are also other cells in the nervous system that we call glial cells. So glia translates literally to glue. So previously thought of really just being the glue that holds everything together, that holds the neurons together. But the glial cells do a lot more than that, we're discovering. So the particular type of glial cell that I'm going to talk to you about today are called astrocytes. Astrocytes because of their star-like anatomy. So within our spinal cord, within these neural networks, we have all of these star-like astrocytes that are also in amongst the neurons. Here's an actual image of that. The green blobs here are our neurons, and the red is showing some of the astrocytes that are jammed in, and many of them in between the neurons. So if we come back to how our neurons are working, how do the astrocytes play in? What's their role in this? So if we add astrocytes, the traditional role for astrocytes is to support the neurons. One of the ways they do this is that they actually hoover up the neurotransmitters that the neurons release. So they remove them from this connection we call the synapse, and then they give them back to the neurons to reuse. So this is one of the very key supportive roles of the astrocytes to make sure that signaling can happen again. But we've been looking, and many people around the world have been looking at whether or not astrocytes do a little more than that, and could they be more active contributors to these neural networks where we've normally thought about the neurons doing all of the work. So could the astrocytes actually release signals that directly affect the neurons? And we think that's the case, and I'll talk a bit about that going forward. And we think that actually they do release substances much like neurotransmitters, but we call these gliotransmitters that can affect neurons and their activity. So how have we been studying the role of these neurons and astrocytes within the spinal cord in our control of movements in the lab? This is just an image I thought would be helpful to show what one of our what we call rigs looks like in the lab. So this is the kind of equipment, the kind of setup that we utilize to study these things. So one of the key techniques, the key sort of preparations we use in the lab to decipher the sort of normal role of astrocytes in controlling the spinal cord's activity is an isolated spinal cord from rodents. So this here shows, this image is one of these spinal cords, and you can see these white light projections are the nerves coming off the spinal cord, which would be connecting to the muscles. And you can also hopefully see these other two sort of structures here, which are what we call glass electrodes, which have been attached to the nerves to record the electrical signals coming out from the spinal cord. So here's my stylized version of that. We have this is the lumbar part of the spinal cord, and we have these nerves coming off the spinal cord and these glass electrodes, which we can attach to record activity from the left and the right sides, also activity coming from that would be rather going to flexor muscles or extensor muscles. So we can record the electrical signals that the spinal cord is generating, and we can actually give it a little bit of a kick with an electrical stimulation or a pharmacological stimulation, and we can get this central pattern generator I mentioned, this network that controls walking. We can get it going even in this isolated piece of spinal cord that we've kept alive in the dish. And if we do that, we see this kind of activity. So we get these rhythmic bursts of activity that alternate between the left and the right sides, and also between flexors and extensors. So this would be controlling walking. We can also look a little closer, and we can record from individual neurons within the spinal cord to try and understand what they're doing and how they are contributing to this activity. And so if we look at single neuron recordings, we see these so-called action potentials, these little spikes of electrical signals that they generate. So we can try and understand which types of neurons are doing which roles within this network that's controlling walking. Another really powerful tool we have is we can visualize the activity of the neurons. So we can use special chemicals that when they're inside the cells will give off light only when the cells are active. So we can visualize the activity. We call this live cell imaging. And so here's a wee video of that, and the lighter colors are the active cells, and if you look, you hopefully see some little flashes of light. There are a couple which are some new neurons becoming active. So that's a really powerful way to study the activity of multiple cells all at the same time, but at that individual cell level. So all of these tools enable us to try and interrogate the system and understand how does it do what it needs to do. So we've been studying, as I said, the role of astrocytes, moving away from the neurons a little bit. What's their role, if any, in controlling this walking behavior, or this electrical signal that controls the walking behavior? So we can utilize our isolated spinal cord, and then we can utilize this imaging technique where we can visualize the activity of these cells. And we first asked, well, are the astrocytes actually active during this walking activity. And we do that at the same time as stimulating this activity and recording the walking activity from these nerves. So over here, we're looking at that. The top line shows the nerve recording. So this is the output of the spinal cord, and we give some stimulation, and then we get these very rhythmic stereotype bursts of activity, which is our walking activity. These are electrical signals that would be controlling walking. And if we focus in on different astrocytes using this imaging with our microscope, we can see when they're active. And what we see is that the astrocytes become more active during this walking activity. And as scientists, we like to graph things. So here's a graph showing that as we get to the walking activity, we get a lot more activity in the astrocytes. So that's great. The astrocytes are indeed active during this walking activity in this isolated spinal cord. So if they're really playing an important role, we would think if we could manipulate the activity of the astrocytes, we would see some change in the electrical signals controlling walking. So we've also done that over numerous years in different experiments. So what we're assuming here is that the astrocytes are providing a signal to the neurons within the spinal cord to control or regulate their activity. So what we can do is use various techniques, drugs and genetic techniques, to activate specifically the astrocytes, turn up their activity, and hopefully turn up whatever signal they're providing to the neurons. So what happens when we do this? And again, we do this while recording this walking in the dish. So here are examples. We've got our baseline control state. Here's this rhythmic activity, and then we stimulate the astrocytes, and we actually get less activity when we stimulate the astrocytes. And here's a graph of that. As we stimulate the astrocytes, the frequency of these events that are related to walking actually decreases. So what if we do the opposite? What if we apply things that will turn down or block the activity of these astrocytes? What do we see then if we're blocking the signaling between the astrocytes and the neurons? So here's that example. Here's our control, and then we inhibit the astrocytes. We turn them down or off, and now we actually get more activity. And we see that again in the graph. When we inhibit the astrocytes, there's a greater frequency of this walking-related electrical signal. So this is quite interesting. So it tells us that actually the astrocytes are providing some negative feedback to the neurons. So I'll explain that here. So here's our network of neurons that are controlling walking activity, and we have our astrocytes nearby. One thing I've not talked about today is we've also studied and seen that the neurons can signal to the astrocytes, and actually the neurons can drive activity of the astrocytes. And then, as I've shown you here today, the astrocytes can actually feed back to the neurons, but they inhibit the neurons. So we call this a negative feedback loop. So one question is, well, why would we have this? Would this make sense? One of our ideas is that if we were to consider the amount of activity that's being generated by these networks of neurons, these CPG neurons to control walking, what if that activity becomes a little too intense or too great? And there might be a risk of damage associated with that. So we think that in that kind of scenario, the astrocytes then provide this brake on the activity to bring it back down a bit. So we think that the astrocytes, by providing this negative feedback, this braking mechanism, that they might keep the neurons working in the sort of optimal range to control the behavior, but keep them away from a range that might be damaging. So that takes me on to the second part I want to talk about briefly, which is the sort of role of astrocytes in disease. A few years ago, I was trying to explain this to my son, then a primary school kid, and he was asking me what these astrocytes were. So he knew about neurons, being the son of a neuroscientist, but he hadn't heard about astrocytes. So I tried to explain their roles in sort of health and disease, and he stopped me and he said, "Oh, okay, so Dad, that means that astrocytes are like the Lando Calrissians of the central nervous system." So I thought this is quite a good analogy for today, given it's May the 4th. So I said to him, "Okay, you're going to have to explain that to me, Finlay. Take me through this rationale." So he said, "Well, you know, Lando," so Star Wars characters here, Lando Calrissian is a great friend of Han Solo to begin with. He's his friend, he supports him. And then Finlay said, "Well, but then when the disease comes along, or Darth Vader, then Lando turns bad. So under the influence of the disease, or Darth Vader, Lando starts doing bad things to Han Solo. He's no longer a friend, and in fact, in this case, he's now freezed him. So I said that's a great analogy for Finlay. Yeah, I like that. Can I keep using that? And he's agreed that I can, as long as he's cited as he is at the top. So the disease that we're studying and where we're looking for the role of astrocytes is, of course, motor neuron disease. Also goes by amyotrophic lateral sclerosis, Lou Gehrig's disease. You'll hear it under a number of names. There's been many prominent people affected by the disease. Of course, Lou Gehrig, who oftentimes in the US the disease is named after a famous New York Yankees baseballer. Stephen Hawking sadly passed away not too long ago from the disease. Closer to home, we have Dodie Ware, former Scottish rugby player, who's been amazing in increasing awareness and also bringing in funding to support people with the disease and also research. You might also remember the Ice Bucket Challenge a few years ago. This is us with the help of our kids doing that some years ago in the back garden in Scotland, which is a little cold for it. So what's happening in this disease? Well, first of all, let's come back to the sort of neurons involved. So we have obviously motor neurons involved, and we actually have upper motor neurons, which are in the brain, and we have lower motor neurons in the spinal cord. And of course, as we've mentioned earlier, these lower motor neurons in the spinal cord control our muscles and our movements. So in motor neuron disease, unfortunately, both the upper motor neurons in the brain and the lower motor neurons are lost. They slowly degenerate, they slowly die. And of course, that leads to a loss of control of the muscles, so a slowly progressing paralysis. Now, unfortunately, once the disease starts, it's not actually that slow. The average life expectancy from diagnosis is sadly 2 to 5 years, but there's quite a big range actually in that. And the real problem is that we don't have a lot we can do in terms of treatment. So we really need to understand this disease better to be able to come in and design treatments to really properly help people with this devastating disease. So one of the ways that we've been doing this is using human stem cell based techniques. So we're using these human stem cells not to try and replace the neurons that are lost, but rather at the moment to try and understand what's happening to human motor neurons and their nearby neighbours in this disease. So this is a fantastic technique derived from Yamanaka and colleagues who got the Nobel Prize for this some years ago. They discovered a way of taking skin cells from individuals and then making those skin cells turn on certain genes, which turns the skin cells into stem cells. Now, all of the cells in our body start as stem cells. So once we have these stem cells, we can turn them to anything we like if we know the right signals. And it just so happens we know the signals to make motor neurons and to make astrocytes. So we could quite quickly, after this technology was invented, create motor neurons or astrocytes in the lab from these stem cells. So here's what they look like. These are some motor neurons on the top here, and here are some astrocytes that have both been made from these specialized human stem cells. So what we've done then is we've taken some of these stem cell derived astrocytes and we've grown them in the lab, put them in a dish, and then on top of them we've placed some stem cell derived motor neurons that have been grown in the lab. And now we can study from these human cells their electrical signals, their function. And this is these action potentials which you've seen earlier. And what we've done in these studies is we've taken astrocytes that have been made from stem cells that have themselves come from healthy individuals, these would be our control lines, or from multiple different motor neuron disease patients. Or also we can take some of the cells, these stem cells, and do a technique that colleagues elsewhere have helped us with, where we correct the gene mutation in the cells to see if that changes things. I should have said earlier that motor neuron disease is often, well, actually not that often, about 20% of cases they involve some sort of mutation in a gene that's passed on through the family, but actually the remaining 80%, we don't have a family history and there's no obvious cause. So we've put these different types of astrocytes in the dish, and then we've put otherwise healthy motor neurons. They've come from stem cells derived from a healthy individual on top to see if there's a negative influence of the MND affected astrocytes on the otherwise healthy motor neurons. So when we record the electrical signals that these motor neurons can produce, we see different types of signals. Some of them down the bottom here show this multiple action potentials, these spikes of electrical activity. We call this repetitive firing. Some show a few, some only one, and some don't show any of these electrical signals at all. So if we look at this graph over here, we're looking at the different types of electrical signals generated by these motor neurons. So the first bit here is showing healthy motor neurons on healthy astrocytes. And you see that some of them don't fire these electrical signals, and there's a range of different types of signals we get. We then look in the middle at healthy motor neurons grown on the MND astrocytes, and we see again a similar kind of profile. And then on the right, we have healthy motor neurons on these gene-corrected astrocytes, and again a somewhat similar profile. But if we keep these cells going, keep them alive in the lab for longer, so up to 10 weeks, we see that in the motor neurons that are on top of the MND astrocytes, we now get a much bigger blue bar. So many more of these motor neurons are not showing any electrical signals as they should. So this is the no action potentials, the blue bar here. And that isn't appearing in our other cases where we don't have the MND astrocytes. And actually, if we look earlier, we see the same thing even at 5 to 6 weeks, that only in these motor neurons on the MND astrocytes do we see the loss of this normal electrical signal. So that's summarized just briefly here. Here's our healthy motor neuron. We can stimulate it, see its electrical signals. If we then put these MND affected astrocytes with the motor neuron, we see a reduction in the electrical normal signals that the motor neuron is producing that would be needed to control the muscles. And in fact, if we wait longer, we see almost no signals generated by these motor neurons. Now, interestingly, at this point, the motor neurons are still alive. They haven't degenerated yet, but they're unable to serve their proper function. So this, of course, leads to the big question of, well, what is it that's going on here? What's changing? Or what effect are these astrocytes that are coming from patients having on the motor neurons? So in order to understand that, we need to think about what generates these electrical signals in motor neurons. And that's done by things called ion channels. So these are proteins on the motor neurons that allow things like sodium ions to come into the cell and potassium ions to go out of the cell. And it's the movement of those charged ions that actually generates these electrical signals of the neurons. So as we see over here, this sort of healthy train or repetitive firing of these action potentials, these electrical spikes. So what we find is that when we grow these otherwise healthy motor neurons with these MND astrocytes, we get either a loss or a dysfunction of these ion channels. And because we don't have that same flow of ions in and out of the cell, we get this reduction in the activity of the motor neuron. So of course, that leads to the next question, which is, what is it that the astrocytes that are affected by motor neuron disease, what are they doing to change the either expression or the function of these ion channels, these small proteins on the motor neurons? And that's something that we're currently trying to study. Excitingly, we're also just embarking on a new study where we've teamed up with a company called Astrocyte, who is designing new drugs, new compounds that try to target the signaling between astrocytes and neurons, and to try and use that to treat disease. So they're focusing on dementia and Alzheimer's, but also now we're looking at whether these might be useful for motor neuron disease. So at the moment, it would be a question of whether some of these drugs that affect the signaling between astrocytes and motor neurons might alleviate or reduce these problems we see in our motor neurons in the lab that are unable to produce the signals they should. Okay, I'm going to stop there, but just acknowledge the people who have done a lot of this work. So current and former lab members, hopefully I've listed them all. And I've underlined some of the people whose work I have directly shown today. So Matt and David and Emily, for example, did a lot of the work looking at the normal role of astrocytes in the spinal cord and controlling walking activity. Then Anna Claire, Veronica and Amit did some of the work I've shown looking at how the astrocytes are affecting motor neurons in motor neuron disease. And that motor neuron disease work has been done in collaboration with groups at the University of Edinburgh, So big thanks to Siddharth and Chandran's group, who we've collaborated with for a number of years on that work. If you'd like to find out more, I'm happy to receive emails. Here's my email address. We also have a lab website and a lab Twitter account. And really importantly, I want to thank the various organisations that have supported through funding our research. And for the work I've talked about today, in particular, that's Motor Neuron Disease Scotland, the Motor Neuron Disease Association of the UK, the BBSRC, and also the Ewan MacDonald Centre. So I'm going to stop there and I think hand back to David.
Thank you, Professor Miles. So we'll now move to the question and answer portion of the event. As I said, you can type your questions into the chat bar, and then I'll select some out to read out to Professor Miles. My colleagues will also put an event survey into the chat bar as well, which we hope you'll fill out just to give us some feedback on this and improving future events. So thank you very much for that. So a first question to start us off, in terms of the ability to create motor neurons from stem cells, does that suggest that the world can anticipate some sort of gene therapy to treat motor neuron disease?
Yeah, that's an interesting question. I guess the element of gene therapy is probably somewhat could be separate to the stem cell world, and in fact in some forms of motor neuron disease, or something called spinal muscular atrophy, which is another disease in which motor neurons are lost, we already have some successful gene therapy. In that disease, there's a specific type of protein that's not being produced at the right amount in the body, and so some successful gene therapy has actually used viruses to put that gene into the cells and get them to produce more or appropriate levels of this key protein that keeps the motor neurons alive. So there are trials going on for motor neuron disease, for amyotrophic lateral sclerosis as we know it in the literature, particularly for the versions of the disease which involve specific mutations in genes that are passed on through the family. So there's work going on currently to see if there would be ways of correcting those sort of problems in the genes. But I guess the other issue we have is that, this is a disease where 80% of folk, there isn't a clear sort of family history, so we have to sort of think about what other solutions we might have to come up with.
You mentioned there are different types of neurons in the spinal cord and in the brain, but are there also different types of astrocytes?
Yeah, it's an important question that lots of neuroscientists are starting to think about. So yeah, a long time ago we thought of neurons as neurons, and then we realized there were many different types, and I think the same is true of astrocytes, that we're learning that they are different in different parts of the nervous system. And so a major challenge now is to understand the different types of astrocytes and the different jobs they do. Unfortunately, all good science becomes more and more complicated the more you learn, and this is another example of that. So, but I think it's a key area that people are working on now is what are the different types of astrocytes? Because I guess from a disease perspective, we need to be targeting the right type as well, because I'm sure there'll be some that are either damaging or supportive in different situations, and we need to know which ones those are.
And a question on that from Sarah: are there any target genes in astrocytes that you are aware of and can share information on?
Yeah, it's a good question too. So there has been thoughts of getting the astrocytes, for example, to express more of types of proteins that are supportive to the neurons. So there are certain types of proteins called growth factors which help neurons survive. So some studies have looked at trying to essentially force the astrocytes to produce more of these growth factors that might then sort of provide a more healthy or protective environment to the motor neurons, even in the face of the disease process. So that's one example. And I think as we learn more about astrocytes and their roles, we're bound to have more targets that we can focus on.
So a follow-up question from Kat here. Do the motor neuron disease astrocytes permanently affect the ion channels, or is the effect reversible if healthy astrocytes are then introduced?
Yeah, that's a really good question too. We haven't done those experiments directly. We have looked a little bit about if you sort of mix and match, and it does seem to be a kind of tipping point where you have enough of the sort of disease-affected astrocytes to affect these ion channels. But we don't know yet from those kinds of experiments whether we could then reverse it by bringing in the healthy astrocytes. And that's something actually going back to the stem cell treatment that people have looked at in some animal models, is could we improve things by putting in healthy astrocytes? So it's an interesting possible direction, I guess, to sort of up the number of the good guys, if you like.
I'm just going to apologise in advance. This is the first time I've used this location to do one of these talks, and I'm underneath a skylight, and it started to rain really heavily. So apologies for the extra noise. I'll mute my microphone when the professor's talking. Anonymous question now: what types of neurotransmitters are present in hind limb movement?
Right, so there are quite a number of neurotransmitters involved in these kinds of activities. So the motor neurons, which connect directly to the muscles, they use a neurotransmitter called acetylcholine at the junction between the motor neuron and the muscle. But within the spinal cord, we have many different neurotransmitters. A major one that is the sort of excitatory one is called glutamate. We have inhibitory neurotransmitters, which are called GABA and glycine, and there's many others, including things like dopamine that you may have heard of in terms of the brain and reward and addiction systems. Also serotonin, which maybe people have heard of as well, which have effects in the brain, but also the spinal cord. So there's actually a vast number of different neurotransmitters that are used by these different neurons. It's one of the ways in which we can tell the neurons apart. Which type of neurotransmitter are they using?
Just essentially a follow-up question on that is, how do you know what type of cells you are recording from? How do you tell the noise? How do you tell the cell from the noise? Yeah,
it's a tricky one, and actually it's something that people have struggled with for many years, and probably I guess a couple of decades ago or more now, we got a lot of help from developmental biologists who started to study the genes that give rise to certain types of neurons. And once we sort of unpack that, we've then been able to use genetic techniques to sort of tag neurons. So tag them through the development so that they might express a protein that fluoresces. So there's something called GFP, which is green fluorescent protein, which we can make certain neurons express this protein, and then when you shine a light at them, you can see them, so you can reveal them. So a lot of it can be these sort of genetic tricks in, for example, mice or lots of other animals. Sometimes you can tell from size, or you can tell from the types of activities they engage in, but it is quite a tricky piece of the puzzle, is trying to actually define the different sets of neurons, because they don't always they don't line up in a kind of simple kind of pattern typically.
I have a question from Evan here. I was wondering how exactly the motor neurons are eventually killed. Is that due to the defective astrocytes, perhaps via ion imbalance, or is something else going on?
Yeah, that's the question we're trying to work out, I guess, and many people are. And there are multiple pathways that probably converge, one would think. I guess the short answer is we don't know the exact final mechanism by which the neurons are lost, but probably there's a cascade of events. So it may be, in the case of our ideas about changes in the ion channels, that as the neurons become less active, there are also some studies that suggest they're excessively active earlier on, but as they become less active, that might then lead to other pathways within the cell kicking in that lead to what we call cell death cascades. So actually during development, some neurons die, they're lost naturally, and some of those same processes could come about in cells that are no longer active, but there are other features many pathways that we think probably converge to end up in loss of neurons. Interestingly, some of those pathways are shared across different diseases of the nervous system. So there's also the hope that if we find treatments that protect, let's say, the neurons that are involved in Parkinson's disease, that perhaps those same treatments might eventually be useful for motor neuron disease, because there may well be similar kind of later endpoints to disease. So that's the sort of hopeful view of our investigations.
And a perfectly timed question from an anonymous member of the audience. Although this research is aimed at motor neuron disease, do you think there may be a crossover to assist with spinal injuries?
Yeah. So as I think I just mentioned briefly, a lot of the basic work we do, just trying to understand how things work normally, I think is very likely to, in the long run, have benefits for motor neuron disease, but also for things like spinal cord injury. As I mentioned early on, it's intriguing that in some animals you can actually get a lot more recovery after a spinal cord injury than we can. So people are looking at why that is, and can we either reconnect things, or can we sort of patch into the systems that are still working within the spinal cord? If we understand how they work, and we have a really deep knowledge of how they work, can we directly activate them, for example? And people are trying that in some parts of the world, to say, could we directly activate the CPG network in the spinal cord to get walking back in someone for whom the connections aren't there between the brain and spinal cord? So, it's really important we do the basic research alongside the more classic sort of translational biomedical research, because there's so much we don't know about the nervous system. And if we don't have that baseline information, it makes it very hard for us to come up with ways of fixing things.
And Evan Moffat's asking a follow-up question to that. Is it possible that dysfunctional astrocytes also play a role in other neurodegenerative diseases that are also present with motor abnormalities like Parkinson's?
Yep. There is definitely evidence of astrocytes having a role in multiple diseases. I believe in Parkinson's and dementia. So yes, I think whether the mechanisms are identical or not, we're unsure, but there is definitely plenty of evidence that these types of cells can contribute to multiple diseases. In fact, even in spinal cord injury, we have something called reactive astrocytes that can come along and be a problem at the site of injury.
Another anonymous question out here is, do we yet understand the reason for the 2 to 5 year prognosis for motor neuron disease, and has that dial ever shifted because of some treatment pathway?
Yeah, so it's a really intriguing aspect of these kinds of diseases. One thing is that we know the diseases started a long time before diagnosis for many of the neurodegenerative diseases. I think Parkinson's is a good example, where 80% of the cells have been lost before you really see symptoms, and I would expect there's similar things going on in motor neuron disease. There's a lot of the problems have initiated well before we see anything. Why there's a rapid sort of time course, we don't know. There's some sort of tipping point, I guess, at which there is quite rapid loss of neurons in many individuals. But there's there is the variation there. Stephen Hawking is an example of someone who was around quite a long time with the disease. Sadly, we haven't shifted that time course much. One of the approved drugs, the main drug to treat motor neuron disease, is something called riluzole. Unfortunately, it only extends life expectancy in the sort of big average trials by 2 to 3 months. So we're not talking much there. So, I think it's another important thing to think about is, yes, we want to cure these diseases, but also we want to just do better in helping people live longer and live better. And so I think that's a major challenge too. Can we come up with ways of extending life, of improving life, as well as looking for cures?
I'm just going to move to a final question now, again about astrocytes and their effect on other diseases. Are they also implicated in multiple sclerosis?
Possibly. There's another type of glial cell that may be more important, something called an oligodendrocyte, that may be more important in multiple sclerosis. It's actually a type of glial cell that forms a sort of insulation around that axon, that long wire-like projection that allows neurons to talk to each other. And it's actually the loss of that insulation that slows down the signals in multiple sclerosis and causes a lot of the problems. So yes, I think glial cells absolutely again have a likely role to play.
I've just had a final question from Claire, so I'm going to just throw in this bonus question. Great talk, Gareth, thanks. Could this just be a loss of function in the affected astrocytes in supporting the motor neurons surviving in vitro, as the firing also seems to be decreasing in the normal motor neurons? I.e., is it just happening faster?
Could be. So I guess one of the questions there is, do we have a loss of the normal supportive role of the astrocytes, or do we have the astrocytes providing some new toxic signal? We tend to be able to keep the human stem cell derived motor neurons working pretty well for a long period of time. I think we've gone as long as 20 weeks, but we definitely can't get that in a disease state. So it could be that it's a difference in sort of support over time, but yeah, all things to try and work out.
And just to say thank you, Professor Miles, for your time. If you haven't filled out the event survey yet, the link is in the chat, and we'll also be dropping Professor Miles's email as well into the chat. So if you want to continue the conversation, please feel free to do so. And you can also watch this Saints talk. It'll be back on our YouTube channel tomorrow and on our website tomorrow, and you can see previous Saints talks as well, and also follow us on our social media accounts: Facebook, Twitter, LinkedIn and Instagram, and also you should be receiving emails about this. So thank you again. Do fill out the event survey, that's very helpful. Just to reiterate my thanks to Professor Miles for his time and sharing his expertise with us all. Thank you very much, Professor.
Thank you very much. Thanks for giving me the opportunity again.